So how is everyone doing?
Pretty good.
Good.
What'd you guys
Good,
think
how are
about
you?
the exam?
It was okay.
Yeah, I expected it to be worse, so that was good.
Okay. So this was the average between my 3 sessions. Well, not the average, like the range of where students landed. It's a good start. The average for my sections were a C+ range, so that's good. Definitely need to get you guys that are down here more up, but we got time to sort that out. It's still the first exam, so you guys will be fine. Okay. But don't panic. You guys will be fine. You guys have 3 more exams, a bunch more mastering genetics assignments, and then with discussion, you have the bunch more quizzes and the presentation points, which should be a piece of cake. Okay? So just a little bit of a change from what I've been doing. I'm gonna take out the discussion videos just because I recently realized that discussion is a tiny bit ahead of lecture. So I'm gonna make it more like a mix with concepts and problems. But as I've always said, discussion is not a substitute for lecture. So make sure you guys actually look through the concepts there too, okay? Everything else is gonna be the same. The quizzes, they're gonna be online as always. There won't be a password anymore because these videos are eliminated. So you guys can just do that whenever, okay? And this stays the same, obviously. So since it's been a little bit of a crazy week with exams and Lord knows what else, I'm gonna keep quiz number 3 open until tomorrow night, 11:59 p.m. So if you haven't done it yet, please make sure to do them. So quiz number 4, there's no password to it. It's gonna be like a check-in attendance point kind of quiz. You guys will see it's like the easiest thing. Just so I can get an idea as to where you guys are at and like how you guys are doing, and if you guys want to give any feedback on discussion itself, this is the quiz. And I promise there are no wrong answers. So whatever you put down, it's totally fine. Okay? So that will be due next Wednesday, 11:59, the typical. Okay? Any questions before I go on? Yes, no, maybe so.
Nope.
Okay. So now we're gonna go into presentations. So I'm gonna stop my share. So, and let you guys, whoever wants to go first for today.
I'll go first.
Okay.
So I can just press share screen.
Yes, you should be able to.
Oh, wait, sorry. Okay, I think I should be able to do it now. All right, can you guys see that?
Yes.
Yep.
Okay. Okay, so I got question number 1, which states 2 black guinea pigs were mated and over several years produced 29 black and 9 white offspring. Explain these results, giving the genotypes of parents and progeny. So some background information. We'll look at like genetic terminology first. So homozygous, both alleles are the same. An example would be 2 big Ds or 2 little Ds. Heterozygous is when the alleles are different, which would be a big D, little d. It's also important to note that each heterozygous or hybrid individual produces 2 types of gametes with equal frequency concerning the plant trait. I'm not sure why it says height. So it could be big D or little d. And then lastly, it's important to remember that one of Mendel's 3 postulates are when the dominant unit is present, the recessive one does not show its trait. And so the answer that I got was that the ratio of the black to white will be 3:1, indicating that there was a heterozygous by heterozygous mating. Based on the ratios obtained, we can clearly understand that the black phenotype is dominant over the white phenotype in the guinea pigs used. The genotype of the black guinea pigs would be 2 big Bs or a big B, little b, while the genotype of the white would be 2 little b's, which is recessive. The genotype of the black guinea pigs under the experimental conditions of this question would turn out to be big B, little b. So the father would be big B, little b, and the mother would be big B, little b. Therefore, the genotypes of their offsprings obtained are 2 big Bs, 2 big B, little b's, and 2 little b's. Once again, relating back to the 3:1 ratio. And in summary, the offsprings with the genotypes big B, and big B, little b will be black, and the offsprings with the genotype with 2 little b's will be white. Yeah.
Good. Everyone good with this question? Any questions?
No. Should I stop sharing my screen?
Yes.
Okay.
I'll go next.
Okay.
Can you guys see?
Yes.
Okay, so my question was, a recessive allele in tigers causes the white tiger. If 2 normally pigmented tigers are mated and produce a white offspring, what percentage of their remaining offspring would be expected to have normal pigmentation? So my information was, in order for a recessive allele to be seen phenotypically, both alleles should be recessive. For example, the normal pigmentation allele is W. That's just an example. That's not the real thing. And the white recessive allele is little w. If a white tiger is born, the genotype must be WW, 2 small w's. If a tiger of a normal pigmentation is born, then the genotype must be either big W, little w, or 2 big W's. Since one of the offspring from 2 normally pigmented tigers are white, the parents are heterozygous. Why? Because in order for a white tiger to be born, they must have 2 little w's. So they must have gotten that from the parents. And to find a percentage of offspring that are normally pigmented, we must do a monohybrid cross with the heterozygous alleles. And here is where it displays the results. As you can see, 3/4 of the offspring are of normal pigmentation, while only a quarter are white. And obviously, if you change that into a percentage, it's 75. So that's the answer.
Good.
All right.
Everyone good with this question? Yes. Great. Okay.
I can go next if that's all right.
Yes.
Okay. So I'll share my screen. And so sorry. Where do I present? Oh, I think it's down here, right? Yeah. Sorry. So my question is, in these disputed parentage cases, the child and mother's blood types are known. The child's blood type is AB and the mother's is A. The two questions that I needed to answer were, what were all of the possible blood types for the father, and what were all of the blood types that would exclude said male from being the father. And the possible blood types for the father would be AB and B. And the blood types that would exclude the male from being the father are A and O. This is because the mother's blood type is A, and the child's blood type is AB. And that means that the father's blood type can't be A, because that would make the child's blood type A or O. It could be AB, because the child's blood type would result possibly in AB. It could be B, and it cannot be O for those same reasons. Yeah, that is my show.
Good. Anyone have any questions before we move on to today's topic?
How do I get back to this? Sorry, I'll stop sharing my screen in just a second. Stop share.
Okay,
All right.
good. Okay, so let me go back to my PowerPoint if it decides to come back. Okay. So now that we know what's happening with Mendelian genetics, okay. So there are some studies that came after Mendel where it kind of continues on with how genes and alleles interact with each other. So there were some extensions and some cases where it defied Mendel's postulates. So in this case, the first thing that was looked in, not the first thing, but it's one of the first ones I'm going to cover with you guys. So, so gene interactions. Gene interactions just mean that a single phenotype can be actually affected by more than one set of genes. So remember how I was talking about like dihybrid crosses where that you'll have this 9:3 to 3:1 ratio unless something else is modifying this, right? So again, we're going to talk about epistasis. That's one of the extensions that we kind of go through with Mendelian genetics. Then the alleles themselves can be either incompletely dominant or codominant, which I'll go into more of that. And then in these genes of different traits can end up not assorting independently, especially when they're on the same chromosome. So if you have like hair color and eye color on the same chromosome, those two might not assort independently because they're linked together like this. We'll go more into that probably next week, I think, depending on how far we get today. And these three types of situations kind of cause deviations from our expected Mendelian ratios of the phenotypes that you see within our offspring. So what is epistasis? Epistasis is when one gene pair affects or masks the expression of another gene pair, okay? So let me just get my little pointer. So epistasis is like this. So this is a regular dihybrid cross, But the thing here is that instead of seeing the recessive type, you end up seeing, so agouti is dominant over everything. Black is like heterozygous, and then homozygous is this guy. So when you have a recessive C allele, that ends up giving it no color, right? So that's when that comes into play. Because if you have at least one of the C's dominant, you'll have some kind of color going on, okay? So in this case, the phenotypic ratio in this epistatic situation, the phenotypic ratio becomes 9:3:4. Does that make sense on why this is occurring? It's because this one gene is affecting the other, that it's controlling the other one. Does that make sense?
Yeah.
Okay. So, oh, if this thing lets me go to the next page, that will help. Okay. So with dominance, we have 3 types of dominance, right? So what we've talked about earlier with Mendelian genetics was complete dominance. That's when one allele or a version masks the other allele completely. But then you have codominance, right? Codominance is when an allele does not mask it completely. So you see a blend of both alleles. Then you see incomplete dominance, where the 2 alleles of the same gene are present and both are actually expressed, okay? Should have added the photo to this, but I do not have the photo on me. Okay. So a question, what term is used to express the idea that several genes exert influence over the same characteristic? What do you guys think?
Is it A?
Not A.
B.
Yes. Okay. Yep, it's B. So we did not talk about pretty much the others other than gene interaction, coadhesion. That's totally unrelated at this point. But yeah, so this is essentially the definition for gene interaction. Gene interaction is when several genes can influence the same characteristic, okay? Okay, so next we have this question where it's saying color of a sunflower pollen can be orange, yellow, or white cream. Crosses reveal that true breeding orange is dominant to both yellow and white cream. However, a cross between true breeding white cream and yellow plants produced F1 plants with a orange pollen. The F2 populations of this cross resulted in a ratio of 9 orange, 4 white cream to 3 yellows. What type of inheritance do these results suggest? So what do you guys think?
A.
Nope. Close though.
B.
Yes. Okay. So essentially with these guys, it really depends on, so from looking at that question, we know that the ratio is a modification of our dihybrid cross, right? So that kind of rules out C through E. So we know it's something of epistasis, right? So to determine this, looking at this, the ratio itself suggests that this epistatic phenotype requires the presence of at least one dominant allele in each gene, right? So in this case, the epistasis will end up recessive, okay? Does that make sense?
Hey, what's the difference between incomplete dominance and recessive epistasis?
Incomplete dominance is, okay, so incomplete dominance is when they're both expressed. Epistasis involves one gene controlling the other gene. Does that make sense?
I don't know.
Okay, let me pull up a picture of codominance, is apparently I forgot to put that in the slide. But hold on. Just give me 1 second. You're on PowerPoint. Okay, I'm just gonna pull out Google at the moment. Hold on. Okay, sorry about that. Let me just pull that up. I'll put this in the PowerPoint. Can you guys see my screen now?
Yeah.
Okay. So here, okay, so complete dominance, when you, so this is seen more in the heterozygous itself. Incomplete dominance ends up with a different color, like it blends the two colors together, right? Did I have it mixed up in the PowerPoint? And codominance is the one that shows both expressed. So you have a blue allele, the color here, but then you also see the yellow as well. I think I had it.
So what's the green?
The green is incomplete dominance. So it's a blend of blue and yellow together.
So do you see like little green spots instead of just blue and yellow spots?
No, you would see it as complete green. You wouldn't see it as spots. You would just see it as green.
Mm.
So with these guys here, you would see the dominant alleles going there. But then when you have them both, these two dominant alleles together, if it was complete dominance, it's completely blue. If it's codominant, you'll have like these guys, like little yellow spots, little blue spots. But then when it comes to incomplete dominance, you'll have it as this. where it's a blend of the two colors or traits. Does that make sense?
Yeah, and then epistasis is how does that look?
Epistasis is more about controlling that color itself, right? Okay, let me just, okay, let me stop this again. And then go back to here. Okay, so with epistasis, so we have AA or anything dominant A is the color, right? So C is the C would be a gene that controls the color itself. So when you have different variations of it, when you have one dominant allele of it, it will show color. But when you see C as recessive, you end up with no color. Does that make sense?
Yeah.
So it's just showing that one gene can control another gene. It can affect or mask the effect that that one gene has on it.
And which one's dominant? Are they both showing or?
So in this case, dominant would be a Gaudi, right? So because like it's not, this is not just about one gene now. This is about two genes.
Oh, okay.
So the gene C is controlling gene A as to how it's being expressed. So with this, so with gene C, it's like it's showing color or it doesn't, right? Because you can have all these different variations of it and still have no color. Does that make sense?
Yeah.
Are we good with the epistasis?
Yeah.
I'm just a little confused on the praxis problem that we did, why it's recessive epistasis and not dominant.
Okay, let me go back to that. So the way that it is given, so since this question is actually saying that, so this ratio here, if it's dominant, then yeah. So in this case, the epistasis would be recessive in the sense that there is at least one dominant allele for this guy, right? Whereas if you, if the epistasis was dominant, you would see no color with C here, right? And then you would just have a lot of white mice instead of the actual this ratio. Does that make sense?
Yeah.
So in this question, it's the similar thing with this scenario where it's still at that 9:3:4 ratio, right? So in this case, it's saying that there is at least one dominant allele in each of the genes. So that's why it's recessive epistasis. Does that make sense?
Yeah. Thank you.
Awesome. Okay. Oh. Yes, okay. A condition in which one gene pair masks the expression of a non-allelic gene pair is called?
B.
Yay. Okay. So that's just a definition question. It's another definition for you guys. What is the difference between incomplete dominance and codominance?
A.
Yes. Does that make sense to everyone? The wording? Okay.
Yes.
With incomplete dominance, a likely ratio resulting from a monohybrid cross would be what? So we can look at genotypic ratio or phenotypic, depending on.
E?
Yes, it's a 1 to 2 to 1 ratio. Does that make sense?
Yeah.
You see different phenotypes. The heterozygous ends up with two heterozygous, one, and okay. Good. What about this guy? In the ABO blood types, all of the following offspring are commonly possible except blank.
B?
Yes. Does that make sense to everyone?
Yep.
Okay. So sex determination. So it depends on the chromosomes you have, right? So we Drosophila fruit flies and humans have like XY chromosomes for males and XX for females. With homozygous, it's homozygous for females, but then you don't say heterozygous for sex chromosomes. You would say hemizygous for males, okay? Just some little key terms to think about. Okay. So X-linked. X-linked, X-linkage, it depends on the presence of genes on the X chromosome. It exhibits patterns of inheritance that is different from autosomal genes. So I'll show you guys an example later how you can determine that. And okay, so the Y chromosome, it's relatively inert genetically. It's male-specific genes on the Y chromosome for the humans, lacks copies of genes that are found on the X chromosome, okay? So let's do a question, if this thing. The inheritance of a skin condition in humans. Consider the following family history. Bob has a genetic condition that affects his skin. Bob's wife, Eleanor, has normal skin. No one in Eleanor's family has ever had the skin condition. Bob and Eleanor have a large family of their 11 children. Of all six of their sons have normal skin, but all 5 of their daughters have the same condition as Bob. Based on Bob's and Eleanor's family history, what inheritance pattern does the skin condition most likely follow?
A?
Yes, but why? So if you guys haven't seen it easily enough, so we have Bob, we have Eleanor, there are 11 children. It's wild at the thought, but 11 children. So we know that we have an affected father, but a normal mother with no history of this disease. And it says that we have none of the sons are affected, and all of the daughters are affected. So when it's going from father to daughter alone, or mother to son, that's when you should think excellent, right? It should be screaming excellent at you at that point. And it shows up right in the next generation. So it's dominant. That's how we end up with the answer as X-linked dominant. Does that make sense?
I have a question.
Yes.
So what would have to happen for it to be X-linked recessive?
That is actually my next question.
Okay.
So we'll go over that too. Okay. So Duchenne muscular dystrophy is caused by a recessive X-linked allele. A man with this disorder would have, what happens? Like, what would be his background?
B.
B.
Yes, you guys are amazing at this. I like it. Okay, so we know that the man has the X chromosome from his mom, right? Because we know that this Duchenne muscular dystrophy is a recessive X-linked allele, right? So the man with the disorder would only get it on his X chromosome. And the X chromosome comes from his mother. And if the mother is not affected, she's a carrier in cases of recessive X-linked allele. So if you had a question like, if this said, all 5 of their daughters have, instead, let's just say this wasn't written here. No one in Eleanor's family has that condition. They could say like, Eleanor's mom had this condition, but we didn't have it in Eleanor. Eleanor would be like, okay, so all of their daughters are carriers of this trait. So when you see that, it could be X-linked recessive if they're showing it from both. So
Okay, so
they were, what's happening? Are we okay?
yeah.
Does that make sense? With X-linked dominant, X-linked recessive?
I have a question also kind of again.
Okay.
So it's X-linked dominant since they only needed to inherit one X chromosome with that condition to show it, right?
Mm-hmm.
Okay.
So with X-linked dominant, you just need one of those alleles to show it, okay? Whereas recessive, you need both of them, or they end up carriers of that trait.
Okay.
And then the carrier women end up giving it to their sons, which automatically becomes that, because there's no other gene that's blocking it. Does that make sense?
Yeah.
Okay. Good. Love it. Okay. So pleiotropy is the expression of a single gene with multiple traits, okay? So it's like one gene giving rise to many different traits. So in this case, this gene is giving out trait A, trait B, trait C. Does that make sense? So this think I took out of the slides themselves. This is an example. So porphyria variegata, it's an autosomal dominant disorder resulting in toxic buildup of porphyrins in the body. Porphyrins are cyclic aromatic compounds that are precursors for heme. So in this case, the genes that build up these guys, when it comes to a point where it's just building up everywhere, it ends up with different phenotypic effects like abdominal pain, muscular weakness, fevers, so on and so forth. Does that make sense with the idea of pleiotropy?
Yeah.
Okay. Good. So the next two things we have is called sex-limited inheritance versus sex-influenced inheritance, okay? So sex-limited is specific to one sex. Like, it's either female or male. There is no if, buts, and ands about it. Whereas sex-influenced inheritance, it depends on the sex of that individual influences it. So it's not limited to one sex or the other, okay? So an example to this guy is like these chickens. So the feather plumage is caused by the autosomal allele. So with the feathers, so it's controlled by this dominant H allele. And this guy, the rooster's feather, is controlled by the recessive allele, okay, which is only expressed in males. So you can see the sex-limited inheritance. You get a specific type of feathering for female chickens, whereas the male chickens get another type because they get their own version of that allele for making it, right? And then the expression itself can be modified by the sex hormones that are being produced in them. So if you see, so in this case, the dominant hen feathered, if you see, for that, even with males, it would be hen feathered, but then with the recessive, it ends up requiring a lot of the male hormones to get that specific phenotype that you see. Does that make sense?
Yes.
Okay. So the next one is sex-influenced inheritance. So in this case, an example would be male pattern baldness. So in this case, we have the big B allele leading to baldness. Heterozygotes end up with baldness in males, but not females. But you can still see it in women if it's homozygous dominant, okay? So it depends, it doesn't matter which sex the individual is, as long as it kind of shows this pattern, it would be sex influenced. Does that make sense?
Yeah, so baldness is dominant and as an allele.
So this is just one example. So in this case, the dominant allele would lead to baldness, and then you would have like heterozygotes having different phenotypes where the female won't end up bald, whereas the males would end up bald.
Is it really just one gene?
That I'm not entirely sure of. This is just one example of how this works, but the topic of sex-influenced inheritance is typically based around one gene. Does that make sense?
Okay.
Okay, so that is pretty much what I have for you guys today. Do you guys have any questions? Oh, and yeah. Any questions, concerns?
Well, the quiz will be open until tomorrow, and it's based on this discussion.
So
It's the same.
quiz number 3 is the one that will be open until tomorrow. Quiz number 4 will be open until next Wednesday. And that's actually an attendance kind of quiz. It's just kind of checking in with you guys, see what you guys need, like just to get some feedback as to how discussion is kind of going. And depending on how that ends up, we can continue on. Does that make sense? Are we good?
Okay.
Yes.
Okay. So I will see you guys next week.
Thank you.
Yep.
Thank you.
Not a problem.
Thank you.
Yep.
Thanks. Have a good week.
You too.